Hello guys, today in this video lecture I'm going to explain glycolysis. So glyco means sweet or sugar and lysis means splitting. Glucose is a 6 carbon molecule, and in glycolysis glucose will be split apart into a molecule of pyruvate, and each molecule of pyruvate represents a 3 carbon molecule. And basically you take glucose and break it into 2 molecules of 3 carbon atoms, and that's the basic idea behind glycolysis. And in this process some of the energy that released as glucose convert into 2 molecules of pyruvate is captured in the form of ATP and NADH. So glycolysis is an energy releasing process. Now let's discuss the net reaction of glycolysis. So glycolysis takes place in the cytosol of the cell. In the net reaction we're going to start with the molecule of glucose, which will react with the 2 NAD plus molecules and 2 ADP units or adenosine diphosphate with 2 inorganic phosphate ions, and this is going to produce 2 molecules of pyruvate plus 2 molecules of NADH plus 2 hydrogen ion will form, and 2 units of ATP will be produced, and then we also get 2 water molecules. So that's the net reaction of glycolysis. Now glycolysis can be broken up into 2 phases: first phase and second phase, and it occurs in 10 steps. So in the first phase, which represents step 1 to step 5, this is known as investment phase. And the second phase, which occurs from step 6 to step 10, known as the payoff phase. So in the first phase we consume energy, and in the second phase we will produce energy, but more energy than what you put in. So in the first phase we need to put in 2 molecules of ATP to get the process going, and in the second phase you're going to get 4 molecules of ATP. In addition, you also going to get 2 molecules of NADH. So that's give us net gain of 2 molecules of ATP and 2 molecules of NADH. So now let's discuss the reactions of glycolysis. Glucose is freely permeable to liver cells. Insulin facilitates the uptake of glucose in skeletal muscles, cardiac muscles, diaphragm and adipose tissue. So glucose is then phosphorylated to form glucose 6-phosphate. This reaction is catalyzed by the enzyme glucokinase and hexokinase along with the hydrolysis of ATP molecule. And this is an irreversible reaction dependent on ATP molecule. And the enzyme hexokinase is present in almost all the tissues. It catalyzes the phosphorylation of various hexoses like fructose, mannose, etc. And this enzyme is inhibited by glucose 6-phosphate. While glucokinase is present in liver, catalyzes the phosphorylation of only glucose molecule and is not inhibited by glucose 6-phosphate. So glucose 6-phosphate is a central metabolic molecule with a variety of metabolic fats like glycolysis, glycogenesis, gluconeogenesis and pentose phosphate pathway or hexose monophosphate shunt. When glucose 6-phosphate undergoes isomerization to give fructose 6-phosphate in the presence of enzyme phosphohexose isomerase. Then fructose 6-phosphate is phosphorylated to fructose 1,6-bisphosphate by the enzyme phosphofructokinase or PFK1, and this is an irreversible and a regulatory step in glycolysis. Then the 6-carbon fructose 1,6-bisphosphate is split into two 3-carbon compounds, glyceraldehyde 3-phosphate and dehydroxyacetone phosphate, with the help of enzyme aldolase. So the enzyme phosphotriose isomerase catalyzes the reversible intent conversion of glyceraldehyde 3-phosphate and dehydroxyacetone phosphate. 2 molecules of glyceraldehyde 3-phosphate are obtained from 1 molecule of glucose. Then glyceraldehyde 3-phosphate dehydrogenase convert glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, and this is an important step as it is involved in the formation of NADH and a high energy compound 1,3-bisphosphoglycerate. Then the enzyme phosphoglycerate kinase acts on 1,3-bisphosphoglycerate, resulting in the synthesis of ATP and formation of 3-phosphoglycerate. So this is a good example of substrate level phosphorylation. If you don't know substrate level phosphorylation, so remember, in substrate level phosphorylation, ATP is synthesized from the substrate without the involvement of electron transport chain, and then 3-phosphoglycerate is converted to 2-phosphoglycerate by the enzyme phosphoglycerate mutase. So remember, this is an isomerization reaction, and then the high energy compound phosphoenol pyruvate is generated from 2-phosphoglycerate with the help of enzyme enolase. So in the last step of glycolysis, the enzyme pyruvate kinase catalyzes the transfer of high energy phosphate from phosphoenol pyruvate to ADP and leading to the formation of ATP. So this is also an example of substrate level phosphorylation, and the reaction is irreversible. Under anaerobic condition, or if there is no oxygen, then pyruvate is reduced by NADH to lactate in the presence of enzyme lactate dehydrogenase. The NADH utilized in this step is obtained from the reaction catalyzed by glyceraldehyde 3-phosphate dehydrogenase. The formation of lactate allows the regeneration of NAD+, which can be reused by glyceraldehyde 3-phosphate dehydrogenase, so that glycolysis proceeds even the absence of oxygen to supply ATP. The occurrence of uninterrupted glycolysis is very essential in skeletal muscle during strenuous exercise where oxygen supply is very limited. Glycolysis in the erythrocyte leads to lactate production, since mitochondria, the center for aerobic oxidation, are absent. Brain, retina, skin, renal medulla, and gastrointestinal tract derive most of their energy from glycolysis. Now let's count out everything that we generated from glycolysis. So first, the glycolysis we know occurring where? In the cytoplasm of the cell. So the next thing that we mentioned earlier, the substrate or the starting substrate of glycolysis is glucose. Now we should know what's the end product. So the end product is going to be 2-pyruvate. Now we understand we get 2 pyruvate out of this process. So we also get total of 4 ATP. Out of these 4, I also use 2 ATP at the beginning of glycolysis, so in a net I get 2 ATP. So what's the next? I also generated total of 2 NADHs. So the last thing that I want to mention is this is an aerobic process. So generally if there is no oxygen, then we will generate lactic acid. So if that happens, what can you generate? You can generate lactic acid through fermentation process. So in short, that glycolysis occurring where in the cytoplasm of the cell your starting substrate is glucose, your end product is gonna be 2 pyruvate molecule. What you're gonna have gross in 4 ATP, but you use 2 out of 4 ATP in this process. So in a net you only have 2 ATP. You also generated 2 NADHs, and most of the times there is very little oxygen. So if there is very little oxygen, then the pyruvate will get converted into lactic acid.